Let's talk about cellular reproduction. Hello there, my name is Timothy Viero, and I will be teaching you about two key biological concepts: mitosis and meiosis. Mitosis is used for almost all of your body's cell division needs. It adds new cells during development and replaces old and worn-out cells throughout your life. The goal of mitosis is to produce daughter cells that are genetically identical to their mothers, with not a single chromosome more or less. Meiosis, on the other hand, is used for just one purpose in the human body: the production of gametes, sex cells, or sperm and eggs. Its goal is to make daughter cells with exactly half as many chromosomes as the starting cell. So let's expand a little bit on mitosis. Mitosis consists of 6 stages, technically 5 if you don't include interphase. It goes through interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. As I mentioned earlier, mitosis is a form of cellular division that produces 2 diploid daughter cells with identically genetic information. Let's talk a bit about each of the stages of mitosis and what exactly goes into going from one single cell to 2 diploid daughter cells. First is interphase. Cells spend most of their time in what's called interphase, and during this time it grows, replicates its chromosomes, and prepares for cellular division. Next is prophase. In early prophase, the cell starts to break down some structures and build others up, setting the stage for division of the chromosomes. And in late prophase, the mitotic spindle begins to capture and organize the chromosomes. Now on to metaphase. The spindle has captured all the chromosomes and lined them up at the middle of the cell, ready to divide. Next up, anaphase. In anaphase, the sister chromatids separate from each other and are pulled towards opposite ends of the cell. In telophase, the cell is nearly done dividing, and it starts re-establishing its normal structures as cytokinesis takes place. Cytokinesis is the division of the cytoplasm to form 2 new cells. When cytokinesis finishes, we end up with 2 cells, each with a complete set of chromosomes identical to those of the mother cell. The daughter cells can now begin their own cellular lives, and depending on what they decide to be when they grow up, may undergo mitosis themselves, repeating the cycle. That concludes mitosis, but what about meiosis? Meiosis in humans is a division process that takes us from a diploid cell, one with 2 sets of chromosomes, to haploid cells, ones with a single set of chromosomes. In humans, the haploid cells made in meiosis are sperm and eggs. When a sperm and egg join in fertilization, the 2 haploid sets of chromosomes form a complete diploid set, a new genome. Meiosis has a 2-step division process. Homologous pairs separate during the first round of cell division, called meiosis I. Sister chromatids separate during the second round, called meiosis II. Starting with interphase again, it's the exact same as mitosis. The cell prepares for cellular division. In prophase I, homologous chromosomes pair up and cross over to exchange fragments. In metaphase I, the homologous pairs of chromosomes line up at the metaphase plate. During anaphase I, the homologous chromosome pairs separate to opposite ends of the cell while the sister chromatids stay together. In telophase, the chromosomes arrive at opposite poles of the cell and prepare for division. Finally, cytokinesis divides the cell, forming 2 haploid daughter cells and ending meiosis I. Now it's time for meiosis II. Cells move from meiosis I to meiosis II without copying their DNA. Meiosis II is a shorter and simpler process than meiosis I. The cells that enter meiosis II are the ones made in meiosis I. These cells are haploid, or have just 1 chromosome from each homologous pair, but their chromosomes still consist of 2 sister chromatids. In meiosis II, the sister chromatids separate, making haploid cells with non-duplicated chromosomes. Now let's get into the process of meiosis II. Starting in prophase II, the chromosomes condense and the nuclear envelope breaks down. In metaphase II, the chromosomes line up at the metaphase plate. For anaphase II, the sister chromatids separate and are pulled towards the opposite ends of the cell. Telophase II, nuclear membranes form around each set of chromosomes, and the chromosomes decondense. Finally, cytokinesis splits the chromosome sets into 2 new cells, forming the final products of meiosis, 4 haploid cells in which each chromosome has just 1 chromatid. And that concludes the full process of meiosis. Now that we know about the processes of both, what exactly are the differences between meiosis and mitosis? Well, here's a list of some basic differences and similarities between the two. Considering the differences and similarities, the basic commonality of mitosis and meiosis is that they both involve the cells dividing and forming new cells, while the main difference of the two is that mitosis results in 2 identical diploid daughter cells, whereas meiosis results in 4 haploid sex cells. So, after all of this, what exactly is the purpose of mitosis and meiosis? Well, they're involved in transmitting genetic information from one generation to the next during the life cycle of animals and plants. These processes are responsible for how that scratch on your hand healed, and also the creation of you! Thanks so much for watching and listening, and I hope you learned something.